Organelle Segregation during Cytokinesis by John Page. Cytokinesis is the process by which a mother cell divides into two daughter cells. Chromosomal segregation is a tightly regulated process of the cell. This ensures that each daughter cell gets the exact same number of chromosomes. This is critical for the continuation of the species. In contrast, however, organelle segregation is not a tightly regulated process, and the number of organelles that each daughter cell gets is a mostly random process. There are, however, some differences between different types of organelles. The first two organelles we are going to look at are ribosomes and lysosomes. Ribosomes and lysosomes move freely around the cell. When cytokinesis occurs and the cell begins to divide into two daughter cells, some of each are trapped in each daughter cell. The number is not necessarily even, and there may be an imbalance. During the growth phases in each daughter cell, the number of lysosomes and ribosomes is replenished to that of the pre-division levels. Next, we will talk about mitochondria, the powerhouse of the cell. Like the ribosomes, mitochondria move freely around the cell. In mammalian cells, mitochondria are localized to the cleavage furrow by microtubules. The cytoskeleton disassembles and cytokinesis occurs. Some mitochondria are trapped on each side of the cell. Mitochondria contain their own DNA and replenish themselves in response to the energy needs of the cell. Chloroplasts behave like mitochondria, but they do not localize to the site of cleavage. Once the cell divides, chloroplasts are replenished. In cells with relatively few chloroplasts or mitochondria, they divide before cytokinesis to ensure that the daughter cells have enough to function. Next, we will talk about the nucleus and cytoskeleton. During cytokinesis, the nucleus and cytoskeleton are disassembled. Once cytokinesis is complete, the constituent parts, which are divided among the daughter cells, are reassembled into the cytoskeleton and nucleus. The nuclear envelope reforms around the chromosome. The final organelles which we are going to talk about are the endoplasmic reticulum and the Golgi apparatus. These organelles function similarly to the nucleus and cytoskeleton. They are disassembled during cytokinesis and dissolved in the cytoplasm. They are distributed evenly amongst the daughter cells. Once cytokinesis is complete, the Golgi apparatus and endoplasmic reticulum are reassembled. The cell then adds membrane to the Golgi and the ER as needed by the cell. Final recap. The division of organelles during cytokinesis is mostly random. Some, such as the Golgi and the ER, are disassembled during cytokinesis, while others, such as the ribosome, stay together. Homeostasis is maintained because the daughter cells are able to function with the amount of organelles that they get until they are able to replenish their numbers to pre-division levels. Thank you for coming to my cell talk.